vaccines. Okay. So when the vaccines when the H1N1 vaccines H1N1 epidemic started, we prepared vaccines for that, but we never used them, so it ended up with a lot of significant loss. So this is why what made the difference is two things. First of all, we have learned a lot about coronaviruses from MERS-CoV and SARS. And second, we had the coalition of empiric preparedness, which means that many foundations, including these, have probably conglomerated together to help speed up the production of vaccines. So if we want to, in routine time, in traditional times, in order to produce a vaccine, we need to determine the target. We know that already the target has been determined from prior studies against SARS and MERS-CoV. Then we should go in a linear fashion to end up with a large production and manufacturing scale. However, during the pandemic, we needed to be as hasty, as fast as the pandemic speed. So since the target was already determined, we, instead of going linear, we went parallel, and this at the risk of losing a lot of money, but luckily many funds were allocated for that. And if we look at what happened usually in the SARS pandemic, so this is the traditional approach. So this part was really relatively done quickly because we know the initial targets. And these phases, instead of doing them after each other in a linear fashion, we did them in a sequential fashion, in a parallel way. And look at the production line, large-scale production line. The large-scale production line was always at the end of all of these steps. Now the production lines were even prepared, and a lot of money was paid even before we get any regulatory approval from FDA, EMA, or any other organization. So are the current vaccines effective and safe? Of course, according to WHO, we know that in order to approve or to endorse a vaccine, we need the approval of regulatory authorities such as the FDA, the European Medicine Agency, the national and regional regulatory authorities in each country or each region, WHO endorsement of the manufacturing firm of the biological product itself. However, for the first time in history, we had emergency use authorization instead of full-fledged licensing. So this is the first time that it has been issued for vaccines. We have used to issuing emergency use authorization, emergency authorization for use for medicines, but not for vaccines. We know from prior SARS and from the current SARS-CoV that the S protein, the S1 subunit spike protein, should be a good target vaccine because it elicits a neutralizing antibody. However, other targets such as the M protein can be also a target for potential future vaccines. So, irrespective of the vaccine, all of them have one concept in general, which means exposing the body to molecules from the target pathogen to trigger a significant immune response and so probably mimicking a natural infection initially. The traditional vaccines prepared by either live attenuated, inactivated, or a subunit vaccine, or a polysaccharide conjugate, which is another subunit vaccine, or toxin or virus-like particles were utilized in COVID. However, a newer technology as the recombinant vector, the viral vector, mostly an adenovirus, and the nucleic acid vaccines, mainly messenger RNA or replicons or even a DNA vaccine, were also utilized. So this is all the vaccine platforms used in producing a SARS-CoV-2 vaccine. We know that we were targeting either the S protein or the matrix protein. And we know this is the most important component of the S protein, the receptor binding domain. And we can usually target, we can have that vis-a-vis inactivated, like attenuated recombinant, or only recombinant receptor binding domain, or virus-like particles, or replication of incompetent vectors, or replication of competent vector vaccine, and of course inactivated virus, or a DNA virus, or even the messenger RNA virus. As of September 17, 2020, 36 vaccine candidates were under clinical evaluation, and 146 candidate vaccine were in preclinical evaluation. So you can appreciate the immense of effort and science that have been put there. And let's see some of the current vaccine, the recombinant viral vector, which are mainly adeno, as I said. This is the adeno 5 coronavirus type 5 vector antigen spike protein. This is mainly made by the Chinese, and this is mainly made by the British, by the Oxford and AstraZeneca, the simian adenoviral vaccine. The unrelated virus engineered to encode for the targeted gene. The advantage is it induces high cellular and humoral response, so it have a very global response. The disadvantages is the presence of possible pre-existing immunity against the adenovirus. So this is why the Oxford people use the simian adenovirus, or risk of reversion to virulence, because they are what you call defective viruses. They have been rendered defective, and limitation in scaling up production. The second modality, the inactivated vaccine, they are mainly Chinese vaccines, and they are easy to prepare, safe. However, they have really quite variable efficacy. But the fact that you are using an inactivated or a killed virus does not mean that probably you may not have really a very significant immune response initially, and you may really need to use several doses. The subunit vaccine, it's also another Chinese vaccine, although there are non-Chinese vaccines. The advantage is safety and ameliorability, scalability. You can, however, it has a high cost, low immunogenicity. We may need adjuvant, and we may need really repeated doses. The messenger RNA vaccines, we have so far two vaccines, the Pfizer vaccine and the Moderna vaccine. They are easy design, rapid manufacture, and they really induce very strong immune response. We need to encapsulate a messenger RNA, otherwise it becomes unstable, and reactogenicity is one problem, and we are seeing it after we have started using this vaccine on larger scales. The DNA vaccines, we have several candidate vaccines here. They have easy design, rapid manufacture. However, they may require special approach to administration and may require adjuvant, and we are not sure about the safety issue. So the Pfizer vaccine was the first approved vaccine, and it's now reaching our country, our region. This is the final data that was presented to the FDA and is now present in the New England Journal of Medicine. You can see here that we have two arms, the vaccine arm and the placebo arm. And after 7 days after the second dose, 1 week after the second dose, we had around 95% efficacy because we had 170 cases. 162 cases were in the placebo group and 8 cases in the COVID vaccine group. A severe COVID case only occurred in 1 case in those vaccinated, but this was after the first dose and not after the second dose, and 9 cases in the placebo, those who placebo. So we have a 90 to 100% efficacy across subgroups defined by age, gender, race, ethnicity, body mass index, and coexisting condition. And the safety profile showed short-term, mild to moderate pain, injection at injection site, fatigue, and headache. And if we look at this data, this is very interesting after the one dose, and you will know, we know that the second dose should happen after 21 days, and look what happens after 12 days. So we probably have somewhere around 80 to 89 to 90%, 89% efficacy after the first dose, especially at day 12. And this is because this is the placebo group and this is the vaccine group. So even after a single dose, you can really have a very good high degree of efficacy and protect and efficiency. The side effects, although they are definitely more than placebo, yet none of them were very severe, and the use of antibiotics was definitely higher in the vaccinated group rather than the placebo. And even when we go into higher age group, the same result have been shown that those who were vaccinated needed more paracetamols and antibiotics, but no severe reactions. So the CDC would say about the Pfizer vaccine, you can use it except in those who have acute infection. So we have to delay it until recovery. So this means that we can give it even to people who have recovered from the infection. In pregnant, lactating, and immunocompromised individual, you have to make an informed decision. There is not enough data about safety in pregnancy and lactation, but it's not a contraindication. This should be informed decision between the physician and the lady. An immunocompromised individual, they encourage you to use it, although data is relatively limited, but there is enough, but definitely those people will benefit from the vaccine. If you have history of anaphylactic reaction, you should be observed for 30 minutes after vaccination. If you have history of allergic reaction, you should be observed for 15 minutes after vaccination. So the advice for all vaccine centers to have what you call a nurse with some preparation for anaphylaxis in case there is anaphylaxis. And now there is a recommendation that if you have developed anaphylaxis against a certain vaccine, you'd better not take any of these vaccines. And if you have developed anaphylaxis post the first dose, you don't need to take the second dose. Luckily, we know that the first dose could be immunogenic in up to 89%. No other vaccine should be given within 2 weeks of the Pfizer vaccine, so you should delay all routine vaccines. And the vaccine should not be used interchangeably with any other COVID vaccine at this stage, although there is now very interesting data trying to study the interchangeability between Pfizer and Moderna, but so far the recommendation is you start with Pfizer, you continue with Pfizer, you start with Moderna, you continue with Moderna, you choose, we start with Sinovac, you continue with AstraZeneca, you continue with the same. The Moderna vaccine, which is very similar, 94% efficacy against the age 18 to 64, 86% efficacy among those above 65 years. It might provide some protection against asymptomatic infection, which is very interesting. 100% efficacy against severe disease, so non-developed severe disease. The most common adverse events are short-lived, and three vaccine recipient and one placebo recipient developed Bell's palsy. So we need to monitor this side effect and definitely monitor allergic reactions. This is the Chinese vaccine that was given in some Arab countries, mainly UAE and Bahrain. They have initially tried 2 doses and ended up using the smaller dose because of less of the side effects. And if you look at the data for neutralizing antibody and for the receptor binding specific antigen, you will find that they are quite immunogenic, especially after the second dose, And the UAE and the Bahrain were the first countries to approve the Chinese COVID-19 vaccine. It is an inactivated virus, grant full approval and not emergency use authorization it was granted full approval by the UAE and the Bahrain authorities, but not by FDA or EMA. The Chinese state or pharmaceutical company Sinopharm is the owner. UAE announced that the vaccine is 86% effective. The third phase clinical trial were done in UAE and Kingdom of Bahrain, around 40,000. Peru suspended testing because of adverse event. Egypt will start using this vaccine soon. However, we lack published data about the phase 3 trial, although we have published data, as I saw you, about phase 1 and phase 2 trials. So what really is important for us for any vaccine really, the best we hope for usually is to prevent infection. So this is really the best, because if you prevent infection, you can prevent transmission. And if you cannot prevent infection, you can really, if you have an infection, it could be an asymptomatic infection. Also, symptomatic infection may affect the transmission, because we know that asymptomatic people may not be transmitter or may not be as a potent transmitter as those who have symptoms. If you cannot prevent disease at all, you should have a mild disease. You should not go to hospital or definitely to ICU or die, but so because you're going to increase the burden of disease. So what really we care for is this is prevent disease, asymptomatic disease, or mild disease to moderate disease. RNA vaccines are now licensed. There is a lot of controversy about whether they are potent or not potent, and whether they are safe or safe. We know that they are very potent, but their safety is also significant. We know that they have the advantage of being as potent as being as safe as a live attenuated, as potent as a live attenuated vaccine, as safe as an inactivated and subunit vaccine. So they combine a good immune response and good safety. If we compare them, because people, they say the nucleic acid vaccine makes them really worry, we have to make sure that a messenger RNA vaccine is different than DNA vaccine, because they have minimal genetic construction. They harbor only elements directly required for expression to encode protein. Recombination in a single-stranded RNA molecule may occur in rare cases, but messenger RNA does not interact with the genome, so detrimental genomic integration is excluded. So this is not genetic engineering, as people claim that, or people who do not understand the molecular biology of it. The lack of genomic integration and the fact the messenger RNA is a non-replicative, metabolically decaying within a few days makes mRNA merely a transit carrier of information and a very safe vaccine. It's very flexible. Any protein can be encoded and expressed by messenger RNA. So this is why they can be used for a variety of usages. And the efficacy, they are very efficacious because they do not need to cross the nuclear envelope. They lack MHS haplotype restriction, and they bind to pattern nicoglycer receptor, and they are self-adjuvanting. They don't need adjuvant, and this is why they can be also another component of safety. The only one problem with formulating them is that they can degrade very easily. So we need to complex them. Complexing means to protect RNA from degradation, help enhance uptake by cell, and improve delivery to the translation machinery in the cytoplasm. And complexing can be usually done with lipid or polymers, mostly encapsulated in liposomes. The second new vaccine that is going soon to be implemented is the adenovirus vector vaccine. Why did we choose the adenovirus? Because adenoviruses have small genomes amenable for easy manipulation. Deletion of a crucial region of the viral genome make them replication defective and predictable with reduced and unwanted or adverse side effect. They have very broad trophism, means that they can infect variety of dividing and non-dividing cells. can be grown to higher titer in tissue culture, so we can produce a lot of vaccine. They are thermostable. They don't need -20 or -80 like the RNA vaccines. And vaccine applied systematically or through mucosal surface, although we prefer systemic application. It is very highly immunogenic because it can stimulate all arms, the innate arm, the production of pro-inflammatory cytokines, and help differentiation of antigen-presenting cell. And it can also trigger the adaptive immune response and virus neutralizing antibody. Recombinant RD vaccine have some limitation, pre-existing immunity, because adenoviruses are very common in children. And some data initially in the older studies from South Africa, not for definitely coronavirus, but for other viruses, have had the questionable risk of acquisition of HIV, but it was not really reproduced later. What vaccines are currently or will be soon available? If we look at this slide, we can see that these are all the technology used in producing the vaccines, and these are the percentage of vaccines that are being used with different technology. So RNA-based, 13%, inactivated around 6%, non-replicating viral vector around 12%, and so on. Subunit vaccine around 30%. The immunogenicity and safety of a recombinant adenovirus type COVID vaccine. This is one of the Chinese vaccine. It showed really a very good reactivation, especially when it used to with the 5 to the 10, and this is the basis also of the Johnson and Johnson vaccine, which probably will be one of the few vaccines that we can use once daily. If you look at the reactivity from the and safety, immunogenicity and safety from the Russian vaccine, which uses 2 adeno vectors, we can see that it really induces strong humoral and cellular immune response in adults less than 60 years of age. Neutralizing antibody titers are higher than those reported by vaccine, are lower than those reported by vaccine-based mRNA vaccines and by the simian vaccine, and volunteers elicited same titer of SARS-CoV neutralizing antibody as did people who had recovered from COVID-19. So this is what happened with the 2 vaccines after the 2 dosages, and you can see, compare it to convalescent sera, they are almost comparable, yet slightly lower than the AstraZeneca vaccine. This is an interesting new vaccine, which is the recombinant spike protein nanoparticle vaccine, and this can has 2. They have the recombinant spike vaccine, and they have also the matrix M1 adjuvant induced. So this is going to be probably produced by 2 pharmaceuticals, and as you can see, it needs 2 doses. And if you look at the safety data, you will know that localized reactions are acceptable in different dosages, and ultimately they will choose 1 dose. And the systemic reactions are also non-severe. Very few were severe, and definitely they are much less in the smaller dosages. And if you look at the efficacy data, you will see that, interestingly, if you use, you can see that the efficacy after 2 doses is very interesting, and comparing it to human convalescent sera. And this is also, the first one was against the wild type 2 microneutralizing antibodies. You will find that also efficacy against human convalescent sera is comparable. So safety and immunogenicity of the AstraZeneca vaccine have been tested, and it showed that it has similar immunogenicity against all age group after a booster dose. However, what's interesting in this study is that they found that the smaller dose will probably may carry even a better potential, and the smaller dose, since it was done accidentally, the AstraZeneca people are now interested in doing larger studies to confirm that what they have observed with the small half dose initially, then a large dose, is going to be can be replicated on larger scales in order to reach the 90% efficacy claimed by the virus. This is also immunogenicity of another recombinant virus given in in a smaller viral particle dose and in a larger viral particle inoculum. And as you can see later on, the Johnson & Johnson people decided that they will use two small dosages of 1 and 1 to compare it with a single dose higher volume of particle, viral particle, inoculum of viral particle, and this is probably what the basis for a single dose vaccine that is going to be soon approved for emergency use authorization by the FDA. So which vaccine do we prefer or we can prefer? This is interesting because let's see, each vaccine has its own merit. The AstraZeneca is not approved yet, but hopefully it's going to be approved, emergency authorization use. The storage is at fridge temperature. Efficacy can be 90% in a small and irregular dose, half dose and irregular dose. It needs 2 doses. The Moderna needs 2 doses, 94% efficacy, FDA approved, but minus 20 degrees, so it needs an ultra light chain. The Pfizer, 95% efficacy, 2 doses, needs minus 70, it's FDA approved. I omitted the cost because it varies among countries. The Sputnik data vaccine, they claim 91% efficacy, however, there is no data. 2 to 8%, so similar to the Oxford AstraZeneca. The Johnson & Johnson, we can use it either in 1 dose with a larger viral particle inoculum or with 2 doses. And we still don't have the authorization. And the Sinovac is an inactivated subunit vaccine, 2 doses. According to UAE, it has 86% protectivity. We don't know yet, and it can be stored easily. So who should be vaccinated? Adults above 16 or above 18. But they say that between 16 and 18, you should discuss that with their parents. Children, there is no clinical data about that, although I believe that in the future, they will be included in the vaccination calendar. Pregnant and lactating women, we need an informed decision. We discussed that with their physician and definitely their physician with them. The immunocompromised are encouraged to use it. People with underlying medical condition and elderly are encouraged to use it. In fact, they are our priority. People with vaccine allergies here, they should be really cautioned, and when they use it, they should stay in the vaccination center between 15 to 30 minutes. People with other allergies, the same. People who have had a COVID-19, the WHO and all authorities would recommend vaccinating them again. People who had taken a COVID-19 antibody treatment, the recommendation so far is to wait at least 90 days after receiving the antibody treatment to subject them to vaccination. People with severe allergic reaction after first dose should not get the second dose. Luckily, as I said before, the first dose could be immunogenic enough. Prioritizing target group, who should be prioritized? I'm giving you now our Lebanese decision that we are going to probably propagate soon. We have to take into consideration ethical issues to maximize benefit, minimize harm, promote justice, mitigate inequalities, promote transparency, and definitely a pro-choice or promote choice. In fact, it's even better. Reach out for the target group, selection issues by priority, and determine who are your risk group. So the first line to receive the vaccine earlier are definitely, and then there are people who are going to receive it later and later. So you have to prioritize given the fact that the vaccine will come in gradual amounts. And you have to remember that this is the first time in humanity history, 7.5 billion people all over the globe need vaccines at the same time. How long does vaccine immunity last? It's not yet clear. I know there is the NIH is proposing a protocol for monitoring vaccine immunity. We in Lebanon are proposing also our own protocol. I think all countries will propose their own protocol, but we're going to learn from each other. This depends upon the vaccine itself. This depends upon the recipient of the vaccine. This upon the level of neutralizing and other important antibodies, other immune component, and definitely the immune memory, because we rely a lot of our immune memory. Do I need to wear a mask if I am vaccinated? The answer now is yes, a mask is needed. Why? No evidence yet that the vaccine can prevent transmission, so we need to have more and more data in order to make sure that the vaccine will prevent transmission. Vaccine can prevent disease but cannot prevent infection in 100% of cases. So even if you take vaccine, you may even get a small or you can get a subclinical or a mild infection. So you still need to protect yourself, prevent you from acquiring other respiratory viruses, influenza, common cold, coryza, adenoviruses, and others. Public health recommendation to wear mask during pandemic is important, and you cannot you cannot always stop and ask people why you're not wearing mask. The public health national level, we will probably recommend the wearing of masks until we achieve good herd immunity level, which is probably 70% of the population. And this may take in Lebanon at least till mid next year, 2022. In other countries, it may take till early 2022, but definitely all 2021 is we should wear masks. Will the new variant appearing mainly in the UK affect the efficacy of the current vaccine? The new variant was hastily named VUI 2020-12/01, which means the first variant under investigation, in December 2020. It has the whole 17 mutations recognized, but however, the most important mutation is N501Y in the S protein that binds to the ACE2 receptor, which is the part that usually the domain that through which the virus will go directly into the cell. The mutation to the S protein, the target of the 3 leading vaccines. So all vaccines so far target the S protein. I know some vaccines may target the S protein and the matrix. Vaccines produce antibody against many regions in the S protein, so it is extremely unlikely that a single change would make vaccines less effective. So at this stage, we believe that the current vaccines we have are going to be effective against the new variant, and the current SARS-CoV does not mutate as fast as influenza viruses. So this is why probably some authorities believe that it may take us 2 to 3 or even 4 years for the need to change our vaccine. However, if we need to do so, the current vaccines are easily tweaked if needed. They can easily be remodified. And this is another interesting variant which is associated with increased spread, because we know that the new variant, the UVI 2020-12/01, can be associated with increased spread. And we know that the viruses they need to vary. If they don't vary, they don't behave like viruses, and especially RNA viruses have to change quickly, always to vary quickly, to vary always, to produce mutation always. Otherwise, they cannot sustain themselves. And while producing so many copies, many of these copies are usually useless. However, some of these copies will be rendered useful for the virus, and the most successful copy will be definitely the copy that will stay with us. To conclude, really, any vaccine deployment should be fully licensed and meets all minimal critical criteria. So for us in our country, we need the FDA, the EMA, and definitely the WHO endorsement, which will take place in a couple of weeks. Certain degree of uncertainty regarding age-independent vaccine activity. We have seen that some vaccines are definitely more potent in younger individuals. No substantive differences in vaccine efficacy in various high-risk groups. Non-pharmaceutical interventions are still needed, such as masks, until we vaccinate very high percentage of the population. No clear evidence that the vaccine will impact viral transmission, but if vaccines can prevent infection, they can also prevent viral transmission. Data about population seropositivity is unavailable in many countries, including Lebanon. Vaccine availability may be limited in initial stages. So this is why, although everybody deserves to be vaccinated, we are going to prioritize our vaccines. Thank you. I will stop at this stage and allow what time left so I can take your questions, and I see I have several questions on the Q&A. So I will stop. I will ask
Yes, thank you, Dr. Bizri.
the moderator to lead the questions, and I will answer them. Thank you.
Thank you, Dr. Bizri. That was very informative and interesting. Really appreciate your time and effort for the presentation. I'm going to go ahead and open the floor for discussion. If you have any question, please type it in the Q&A box. We have a question. What is reactogenicity that you mentioned regarding the Pfizer vaccine?
Yeah, this means that usually these vaccines can produce a lot of reaction. And we have seen it now when we start giving the vaccine, and many are producing certain degree of allergy. So we have to be very careful about this possibility. And the allergy is usually exaggerated in the second dose, not in the first dose.
How do you convince the general public that the speed at which the vaccines have become available for use does not adversely affect the safeness of the vaccine?
Well, you see, there is a very big anti-vaccine campaign all over the world, not against COVID, but against all vaccines in general. And this has been triggered in the past due to certain data that was published before. Whether this data was really good data or not, this is not the time to discuss it. So we have to convince them by telling them first of all, be very transparent about the side effect and the potency of the vaccine, and about what to expect if not vaccinated. And we have to promote their choice. But before we promote personal choice, we have really to empower people to be very frank, very transparent, and tell them in layman word what vaccination mean and what not vaccination mean, and to tell them that many people that are using the social media claiming to be authorities in certain diseases are not true authorities, and this is an exaggerated campaign.
How do you determine which vaccine type would work best for an individual?
Well, this is a good question. We cannot determine, but we know that, for example, if somebody is allergic to, let's say, to a certain vaccine, they can use another vaccine. We know that messenger RNA vaccines, although they can develop a certain degree of allergy because of reactogenicity, in the second dose, they are relatively very safe. We know that inactivated vaccines are safe. So we have really to probably tailor it. But you see, first of all, since we are seeking mass vaccination, we have to cover the largest number of people vaccinated. This is why we are going to encourage any type of vaccine that reaches us soon, yet it should be really fulfilling all the criteria, as I said before, FDA, EMA, WHO. Let me tell you, from now being the head of the COVID vaccine task force, we insisted on the vaccines being provided for free, and we insist that all vaccines should be given to all who are on Lebanese soil, irrespective whether they are Lebanese or non-Lebanese. And definitely we insisted on promoting choice. The individual has the right to accept or not.
Is Bell's palsy avoidable, and when do symptoms resolve?
Let me tell you, this is very interesting. I was discussing that with one of our colleagues who have had Bell's palsy before. So maybe if there is Bell's palsy associated with a certain vaccine, I would wait on vaccinating this individual until we have a second vaccine which is not associated with Bell's palsy. For example, I didn't mention that in my presentation. We have seen some Guillain-Barré in the inactivated, in the subunit vaccine that was given in the Arab states and mainly in UAE, although this data was not published. Although, for example, Guillain-Barré with messenger RNA vaccine is extremely unlikely. So here, probably in certain subset of people, you have to individualize. But so far, 3 Bell's palsy out of a large number may not mean much, but there are certain people who have had it or had a family with a strong family history of Bell's palsy. Maybe we should probably try to keep them for a second type of a vaccine.
With the evolvement of the new strain, if this continues, could we reach a point that the vaccine is no more effective and we have to modify it to be effective?
Well, I've answered this, but let me tell you, the RNA vaccine should evolve. Should they have variation? If they don't do that, they are not RNA vaccine. So far we don't have reasons to believe that the current variants will not respond to the vaccine. They should be covered by the vaccine, because the vaccine, when launching antibodies against the spike protein, they are launching antibodies against several domains of the receptor binding domain. So we probably at this stage we have no reason not to believe that they are effective. However, as per a new German study, they say that it may 2 to 4 years in order to establish a new strain, because we have to differentiate in the virological terminology between a variant and strain. So far, we have what we call variants. We don't have new strains.
Any studies comparing efficacy of vaccines to convalescent plasma?
Yeah, this is good. In fact, I didn't show most of the studies because of the time restraint. But in fact, most of them, especially in phase 1 and phase 2, when they wanted to see the antibody titer, the neutralizing antibody titer, the IgG, and the anti-RBD, the receptor binding domain titer, they compare it to convalescent sera. And on all vaccines in use or recommended are either equal or even better than the convalescent sera. So yes, it was compared, and they showed that either identical or even better to the convalescent sera.
What can you tell about the long-term side effects of the vaccine? Can they appear at a longer period after years, for example?
Well, anything is possible, but let's take we have been using vaccines for so many years, and nobody has really proved a serious side effect with vaccine. My belief is that messenger RNA are going to be very safe vaccines, because the RNA is extremely an unstable molecule. Even with complexing it with lipid, it will not stay for long. It will stay for days and decay and will disappear. And the immune response will remain later as an immune memory that will be triggered when we have a stimulant. But ultimately, nobody can swear against that they are definitely for sure. But yes, will be associated with certain questions we don't know yet. But please, You have the right to question, but always let's compare what happens with if we have a vaccine versus if we don't have a vaccine. If you don't have a vaccine, probably in a country such as Lebanon, we need 6 or 7 years to come to achieve a good herd immunity levels, and this cannot be tolerated by any means.
Which vaccine will be used in Lebanon? When will it arrive? And do we have the necessary infrastructure to properly store the Pfizer or the Moderna vaccine?
Okay, thank you for this question. Probably we're going to receive the Pfizer vaccine first. It will be as early as the first week of February. However, we are only committing ourselves to the second week of February. Other vaccines will come soon. You see, the problem with Lebanon that we don't have money. We have to rely on funding either from the World Bank or from other authorities, international authorities, and those international authorities will not loan Lebanon money before WHO endorses these vaccines. So the World Bank now, I'm giving you now inside information, the World Bank has probably approved the concept, so the Pfizer vaccine will be there, and we have put a down payment from UNICEF money to the COVAX platform in order soon to get other vaccines other than the Pfizer, and maybe it's going to be the AstraZeneca vaccine once it is ready and fulfill the whole criteria.
What can you tell about the reinfection with COVID-19? What is the range period to get infected again after being infected for the first time?
This is also another interesting question. In the past, we never really knew what happened in those who are infected. And people thought that the immunization, that immunity will wane quickly. Now we have data to encourage us that the immunity does not wane quickly, and that reinfection rate and reinfection, although it occurs really in very small and limited numbers. You have to remember that we have tens of millions of people infected, yet the number of people who claim to be reinfected are in hundreds. So this tells you that reinfection is not the norm; it's the exclusion. However, we have to keep that in mind. For how long we can keep that immunity, we don't know, but you have always to rely on our memory, and this is why the WHO and all other authorities now are advising that even people who have recovered from COVID-19, they can take the vaccine.
How far are we prepared regarding the cold chain for the mRNA in Lebanon?
Well, thank you, Rima, for this question. The cold chain in Lebanon is present to our astonishment. We know that we have a lot of assets in Lebanon. These assets were not initially declared, yet they were found. We know that we have at least 14 confirmed cold chain fridges, but we can even have more in our research lab, and we're going to get an extra 6 or 7 soon. And so the infrastructure is there, and the vaccination centers are now being prepared, and I don't believe that Lebanon lacks the infrastructure.
Another question from Rima. How long after the closure of phase 3 studies you would be 100% sure that there are no long-term sequelae?
Nobody does that, but we know that there is what we call post-marketing studies or surveillance studies. In Lebanon, we have established now a body that will monitor the vaccine introduction because of two things. We need to make sure that we are giving the vaccine to the right people who deserve it, and we are probably doing it rightly, and we are going to monitor the side effects, and definitely we are going to rely on others monitoring. For us, we have to remember in Lebanon, we're not going to get the vaccine before February, so we have 2 months to observe the United Kingdom, America, Europe, and other and other regions of the world to see whether what kind of side effects they experience.
Our last question for today is, what are the possible long-term side effects of the mRNA vaccines versus the adenovirus vaccines?
Well, the mRNA vaccine, for the mRNA vaccine, nobody knows because mRNA vaccine are relatively new vaccines. We know that they are relatively safe because they are really a disintegrating molecule. As for the adenovirus vaccines, the adenovirus vaccine have been used in prior vaccination, although not on a very large scale, but have been used in prior vaccination for certain killer diseases, and they have proven to be efficacious. So they are not, they are to a certain extent quite safe.
Thank you, Dr. Bizri, and thank you everyone for attending our
Answer
webinar.
one question by our chief pharmacist, by Ulfat. Yeah, sure. About infliximab, I think I think there is no risk of giving an mRNA vaccine in a patient who have received infliximab. But if there is no hurry, I would probably wait until there is some time to pass until the immunity is regained. But immunosuppressed individuals, even severely immunosuppressed individual, can receive these vaccines, and there should be no major contraindication. Thank you.
Thank you. Thank you everyone for attending. You can forward any more questions you have to international@ahcdc.org. I will write it in the chat box for you. And thank you for joining us. You can check the recording in a few days on our website as well.
Thank you.
Thank you.